Hello and welcome to Nikolay's genetics lessons. In this video I'm going to talk about how by analyzing a pedigree we can find a mode of inheritance of the genetic disorder, and also I'm going to talk about inbreeding coefficient. So let's now take a look at this pedigree, and one by one let's analyze if it can be Y-linked genetic disorder, X-linked dominant, X-linked recessive, autosomal dominant, or autosomal recessive genetic disorder. So imagine that we have one variant where we have Y-linked genetic disorder. So that means that we have a defective gene on the Y chromosome. So genotype of the male would be XY, and of the female would be XX. So this is going to be Y-linked genetic disorder. So this is going to be male side, and this is going to be female side. And when we build a Punnett square, we can predict what the genotypes of the progeny can be. And here we would have two normal X chromosomes. One would be inherited from the mother side, another from the father side, and this is going to be female, of course. And here we would have also two normal X chromosomes. Once again, this is going to be female. And here we would have one normal X chromosome inherited from the mother side, and one defective Y chromosome inherited from the father side. Once again, here we have one normal chromosome inherited from the mother side, and one defective Y chromosome inherited from the father side. So as you see, if father would be affected, all his sons also would be affected. So let's now analyze our pedigree. Do we see this pattern? And if this would be Y-linked genetic disorder, the father of this person also would be affected. But we see that this male is normal. Also, all the progeny, male progeny of this person, one, also have to be affected. So all male progeny. We see here that this couple has a son, and son phenotypically normal. So we can say that this is not Y-linked genetic disorder. Let's now consider answer B, X-linked dominant. In this scenario, our male have to be affected, so that means that his X chromosome have to be defective and Y chromosome have to be normal, and female would be phenotypically and genotypically normal. So let's once again build a Punnett square, and according to this Punnett square, as you see, all the female progeny would be affected because this is X-linked dominant genetic disorder. So all the males, all the male progeny of such a cross would be phenotypically normal because they would get the X chromosome from the mother side. So what do we see here? We see that this couple has a female that is phenotypically normal, but according to our Punnett square, all the female progeny have to be affected because they have X-linked genetic disorder, and at least one of the chromosomes would be inherited from the father side. So this is also not our case today, so we can cross out this answer. And next variant, answer C, X-linked recessive genetic disorder. Once again, male would have one defective X chromosome and normal Y chromosome, and female going to be phenotypically and genotypically normal. And this time our picture would be as follows. So here we would have phenotypically normal female who is going to be a carrier. Here we would have also phenotypically normal female who is going to be a carrier because this is X-linked recessive genetic disorder. here we would have all the male progeny who is going to be phenotypically and genotypically normal because they would inherit X chromosome from their mother. And as you see, in this scenario, 100% of the progeny going to be phenotypically normal. Those females would be carriers, but not males. What we see here that this male have to carry this genetic disorder and transfer it to the following generations. So this is also not our case today. We can cross out this answer. Next, answer D, autosomal dominant, and this time we have one allele that is defective and dominant, another is normal, so this is going to be recessive allele A that is normal, and the genotype of the second person would be small a, small a. Once again, I want you to pay attention that this is autosomal dominant genetic disorder. So here we would have small a, capital A, small a, capital A here, and small a here, and small a here. So as you see, 50% of the progeny would be affected with this genetic disorder, and 50% of the progeny would be phenotypically and genotypically normal, and these ratios would be regardless of the sex of the progeny. Let's now analyze our pedigree. Once again, these two persons have to be carriers or have to be affected in our scenario of the autosomal dominant genetic disorder, but we see that these two persons are phenotypically normal, so they must be small a, and of course in this scenario they wouldn't be able to pass this defective dominant allele to the following generation, so we can exclude this variant also. And we left with only one answer, autosomal recessive. In this scenario, genotype of the person 1 that is affected have to be small a, small a. That means, of course, that his parents, who is going to be phenotypically normal, have to be carriers. So the genotype would be capital A, small a, and capital A, small a. So if both parents would be heterozygous, and if they're going to be phenotypically normal, they would have 1 out of 4 chances to have affected progeny. So as you see, 3 out of 4 chances that the progeny would be phenotypically normal, 50% chances that the progeny would be carriers, but also phenotypically would be normal, and 25% that the progeny would be affected with this genetic disorder. So let's now consider that all the outcrossers, that is here, all these persons here would be outcrossers, that is not genetically related to these two persons. So let's consider that the genotype would be capital A, capital A. So they would be genotypically normal. We consider that this is rare genetic disorder. And in our scenario that this is going to be autosomal recessive genetic disorder, person 1 that is affected have to be homozygous recessive, just like this person here in the second generation. That means that the progeny of this couple have to be a carrier in order to pass this genetic disorder to the following generation. he have to be a carrier or have to be capital A and small a. And what are the chances of such an event? Once again, if we have one parent who is going to be homozygous recessive, another one who is going to be homozygous dominant, as you see, 100% of their progeny have to be carriers. So that means that both these children of this couple have to be heterozygous for this trait. Now let's consider this couple. So we have one person who is a carrier, another one who is phenotypically and genotypically normal. And what is the chances to pass this genetic disorder to the progeny? Not disorder, but to pass this recessive allele. So this is going to be capital A here, capital A here, capital A small a here, and capital A small a here. So as you see, the probability that this couple would pass this genetic disorder recessive allele to this person, so this person would be phenotypically normal, but would be carrier, would be 1/2 chances. So 1/2, or 2 out of 4, or 50%. This is probability for this couple to pass recessive allele to this person. And this person have to be obligate carrier in order to be phenotypically normal, but at the same time to be a carrier and have a recessive allele that he can pass to his progeny. And also person number 2 here in this generation have to be a carrier in order to pass this genetic disorder to the progeny. So genotype have to be capital A small a, and the probability that these two people would have affected child would be 25%. So that means that this person also have to be a carrier, because this person looks phenotypically normal, but as we know, he have to pass recessive allele to his progeny, so have to be a carrier. That means that this person here, also those phenotypically normal, have to be a carrier of this recessive allele. And this person may inherit this recessive allele whether from his parent 1 or whether from his parent 2. So as you see, answer E, autosomal recessive, is the correct answer. And 3 persons on our pedigree left without the genotype, so we can say that this can be capital A and blank space, capital A and blank space, because these 2 persons can be with homozygous dominant, heterozygous, but not homozygous recessive. And person number 6 in the 3rd generation can be capital A blank space, because we don't know whether this person is homozygous dominant or heterozygous, but we know for sure that he is not homozygous recessive because he is not affected. Now we are ready to answer the second question and to find inbreeding coefficient. So what is the inbreeding coefficient for this person? Or in other words, what is the probability that this person would have a small a allele that is common by descent with ancestor 1 and ancestor 2 in the 1st generation. And this is very easy to calculate. We just have to use the formula that is 1/2 raised n. And n stands for the number of nodes between person 1 in the 5th generation and the person 1 or 2 in the 1st generation. Now let me use different color for this recessive allele A, And let's find probability that this person would get 2 recessive alleles from the person 1 in the first generation. And here is going to be a pathway. So this person may give this recessive allele to person 1 in the second generation. Person 1 may give recessive allele to the person 2 in the third generation. Person 2 may give this recessive allele to the person 1 in the fourth generation, and this allele may end up in the fifth generation from the paternal side of this affected person. So this is going to be one pathway, and here is going to be the second pathway, that this person also would give this same recessive allele to this son, and this son would give this recessive allele to his daughter, and his daughter would give this recessive allele once again to her daughter, and she would give this recessive allele to her daughter. And here we have double bond, that means consanguineous mating, so mating between two close relatives. Now let's count how many nodes we have here. So 1, 2, 3, 4, 5, 6, and 7. We don't count this person, we just count how many people stands in this pathway. 2 possible pathways where he would get 2 recessive alleles from his ancestor 1 in the first generation. So 1/2 raised 7, and the answer would be 1/128. But this is not the only one variant that this person here would inherit 2 recessive alleles from the person 1 in the first generation. He also have possibility to inherit this recessive allele A from the person 2 in the first generation. So let's once again count what is the probability would be. So we know that this person have 2 recessive alleles, so one at least have to be inherited from this person here. So this is one pathway, and this would go here, here, and end up here. And the second variant would be if this recessive allele go here, 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 and here. So once again, if we would count nodes here, we would have 1, 2, 3, 4, 5, 6, and 7. So that means that we apply the same rule, and we are going to have 1/2 raised 7. That also would give us 1/128. So now, because we have 2 variants how this person may get this genetic disorder, we have to add these 2 chances. So plus 1/128. And the answer here would be 1/64, or decimal number would be 0.016. And this is going to be inbreeding coefficient, and if you need an answer as percentage, we have to multiply this number by 100, or move this decimal point 2 places to the right, and as you see, the answer would be 1.6%. And this is going to be our answer to the second question. And I also want to note that when we calculate inbreeding coefficient, we are finding the probability that person 1 in the 5th generation would be homozygous in locus for 1 allele that he would get whether from the person 1 in the 1st generation or person 2 also in the 1st generation. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.